Welcome everyone. Today we are going to learn about active transport mediated by light-driven pump. Now at first I want to discuss about what is active transport. In my basic facts on transport video, I have mentioned that there are two types of movements occur during the transport of molecules or ions. One is called downhill movement, another is called uphill movement. The downhill movement is actually known as passive transport. The uphill movement is known as active transport. Here, in case of uphill movement, molecules or ions are transported against their chemical concentration gradient. Now, what is chemical concentration gradient? It means there is a difference between the concentration of molecules. Let's suppose this is the outside of a cell and this is the inside. If these molecules are present outside more higher in concentrations than the inside, this is called the concentration gradient or chemical concentration gradient for the molecule or ions. Now, if any molecules try to uh, transport or move from lower to higher concentration from this direction to these directions, then it requires energy because this process is thermodynamically unfavorable. That's why this process is called active transport. So when molecules or ions are transported against their chemical concentration gradient from lower to higher concentration, that requires energy. That's why it is called active transport. Now, cells carry out active transport in three main ways. One is coupled transport. I have made separate video on this. ATP driven pump also I have made this video. Now today we are going to learn about how active transport are performed by light driven pump. Now here the example of proteins that perform these or transporter that perform this light driven pump is bacteriorhodopsin, and this is the structure of a very similar protein called rhodopsin. Now rhodopsin proteins are present in the mammalian eye retina of rod cells, and the structure of rhodopsin and bacteriorhodopsin are very similar. It contains 7 alpha helical helices that traverse the lipid bilayer. So this is the plasma membrane and this is the transmembrane domain of the protein, and here are 7 trans alpha helices. If you count 1 2 3 4 5 6 7, so this is called 7 transmembrane protein. Now bacteriorhodopsin is a light driven proton pump, so it pumps proton when absorbs solar energy or solar light, so that traverses the lipid bilayer as 7 alpha helices. It was the first membrane transport protein whose structure was determined, and this is the patches of purple membrane of bacteriorhodopsins present in archaebacteria. If you zoom this patch of purple membrane, you will find this type of structure. If you further zoom, you will find this, and this is the single bacteriorhodopsin molecule contains 7 transmembrane helices. Now, patches of purple membrane contains bacteriorhodopsins, and these bacteriorhodopsins are tightly packed and arranged as a planar two-dimensional crystals already in the archaebacteria called Halobacterium salinarum. Now, halobacteria, halophilic means salt-loving. So, this archaebacteria, Halobacterium salinarum, has purple patches, and these patches are actually made up of bacteriorhodopsins that are tightly packed and arranged as a polar planar two-dimensional crystal. So for this structural arrangement, it was possible to determine the three-dimensional structure of this bacteriorhodopsin by combining electron microscopy and electron diffraction analysis. This process is called electron crystallography. For bacteriorhodopsins, the structure was later confirmed and extended to a very high resolutions by X-ray crystallography. So by electron crystallography and X-ray crystallography, the three-dimensional structure was actually generated, and these planar two-dimensional crystals actually helps in the obtaining three-dimensional structure by electron crystallography. Now it contains bacteriorhodopsin proteins contains a single light absorbing chromophore retinal, and this chromophore actually gives the protein its purple color. Now retinal is actually a vitamin A in its aldehyde form. It is identical to the chromophore that found in the rhodopsin protein, as I have already mentioned, rhodopsin protein in the rod photoreceptor cells of the retina. Now, in bright light, each bacteriorhodopsin molecules can pump several hundreds protons per second. It establishes by this pumping of hydrogen it establishes an H plus gradient. Now, what is H plus gradient? H plus gradient means chemical concentration gradient. That means when this bacteriorhodopsins pumps protons out of the cells, basically they pump out of the cell of this archibacteria, then the outside of the bacteria, the hydrogen concentration increases, and that creates a H plus gradient. That means the outside hydrogen concentration higher than inside of the archibacteria. So it establishes an H plus gradient across the plasma membrane, which in turn drives the production of ATP by a proton driven ATP synthesis. So this archibacteria contains another type of proteins called proton driven ATP synthesis. Now this protein can synthesize ATP. So these proteins are present in the membrane also. As like the bacteriorhodopsin present in the membrane, this protein also present in the membrane. So by the help of this H plus gradient, this proton driven ATP synthesis can synthesize ATP. So what is happening here, basically bacteriorhodopsin converts the solar energy into H plus gradient, and then this protein actually utilize the H plus gradient to make ATP. Now this is the series of transfer of hydrogen or protons from inside to the outside of the archibacteria. Now retinal, this chromophore is actually bound to covalently to the side chain of lysine 216 of these proteins. So I am going into molecular detailing of these bacteriorhodopsins, how proton pumps occur. Now retinal, this chromophore is attached with amino acid called lysine 216 via sheep base. The proton pathway means how protons transfer from inside to the outside. The proton pathway across membrane includes the side chains of aspartate 96, aspartate 85, glutamate 204, the sheep base, and the water molecule. So protons actually jump from these amino acids of this protein and ultimate to the outside. Okay. Now local environments means here inside the lipid bilayer, local environments gives the two aspartate means aspartate 96 and aspartate 85 remarkably different ionization constant or pK values. Aspartate 96 has a very high pK of approximately 10, so it can serve as a proton acceptor. So when any amino acids has a very high pK or ionization constant, that means it is a proton acceptor. Now aspartate 85 has a low pK of approximately 2, so it serves as a proton donor. Now let's see how this cycle occurs. The mechanism starts with the retinal in the all trans configuration. So basically this retinal has two alternative conformation. One is called all trans and it is 13 cis configurations. When the configuration of the retinal changes, the structure of protein also changes. So absorption of a single photon can excite the chromophore and change its conformation, and that conformational change causes the changes in the structure of the proteins, and that's why it actually can transfer the protons. So the mechanism starts with retinal in the all trans configurations and protons bound to the sheaf base and aspartate 96 at the hydrophobic cytoplasmic end of the proton pathway. That means here already hydrogen ions or protons are actually attached with aspartate 96 and retinal. Now absorption of a photon isomerizes this retinal. Isomerizes means its conformational changes occur to 13 cis configurations. This changes the conformation of the protein and lowers the pK of the sheaf base. As the pK of sheaf base lowers, it actually donates the transfer the protons to the aspartate 85. So from this retinal, the proton is transferred to the aspartate 85, and aspartate 85 then transfer the proton to glutamate 204 present here, and it releases the proton outside of the cells. Now a further conformational change of the protein reorients the sheaf base towards aspartate 96. Now as the protons are actually move away from this inside to the outside, this chromophore or retinal actually reorients the sheaf base reorients towards the aspartate 96 to actually gain one proton. So the pK of aspartate is lower in this situation, in this conformation, so a proton transfers from aspartate 96 to the sheaf base. Now aspartate 96 is reprotonated from the cytoplasm, and the retinal re-isomerizes to the all trans configuration in the preparation for another photocycle. So this is the whole pathway of the proton pumping, and by this way bacteriorhodopsins actually pump the proton.